So we welcome now Fabiana Quaglia, a professor at the Department of Pharmacy of the University of Napoli, Federico, and she will speak about the antiangiogenic biodegradable nanoparticles enhancing the anticancer activity of chemotherapeutics.
So thank you very much, Maria, for the introduction, and thanks to the organizer for giving me the opportunity to share with you some of our research at the Department of Pharmacy at the University of Napoli, Federico II. Today I will talk to you about antiangiogenic biodegradable nanoparticle. Maybe? It doesn't work. Okay, it was okay. So thank you. So the idea is to use nanotechnology in chemotherapy, and as it has been underlined today, it is important to consider that nanotechnology can allow common advantages, such as protect the drug from enzymatic degradation, alleviate solubility issue, which is a key factor for lipophilic chemotherapeutic, control PK of the drug, and alleviate toxicity profile. But it has been also underlined that control of distribution of chemotherapy throughout the tumor tissue and the possibility to target specific tumor elements such as specific cell subpopulation or ECM, or to target metastasis and overcoming at the same time MDR issue is a key advantage. In building nanotechnology strategies for cancer, core shell polymeric nanoparticles have a number of features, of positive features, such as the possibility to use hydrophilic or not water-soluble materials, also ionizable materials, so you can get a fine control of the sides, a control of the structure, the core shell structure. One single drug or multiple drugs can be incorporated in the core of the shell. Surface properties can be tailored, and at the same time, the delivery rate can be controlled too, and you can achieve sustained or pulsed delivery. In so doing, you have to take into account that bioelimination and toxicity or immune response needs to be checked for correct translation. In my lab, we have worked a lot with this kind of polymer. You see here the chemical structure. It's very simple. It is made of polyethylene glycol linked to polyepsilon caprolactone, giving this amphiphilic copolymer. The two moieties are FDA approved. They can form several structure, core shell structure, and allowed mainly a sustained release of drug cargo. Furthermore, hydrophilic lipophilic balance can be suitably balanced, and you can achieve different solubility in organic solvents, so you can play with different preparation techniques. And another advantage is the low melting temperature, which allows you also to find out novel mode of self-assembly of the system. Of course, pegylation is detrimental for cell interaction, and therefore you need the strategy to modify the surface in order to get these targeting properties and encourage nanoparticle entry inside cells. As you can see here, we played a lot with this copolymer, so we have tried to understand the impact of architecture and amphiphilic properties on preparation technique in order to get reliable results in view of a translation of the system. We have delivered lipophilic chemotherapeutic inside the systems and also tried to amplify the response to cancer therapy by combining different therapeutic strategy and mainly light. We have also demonstrated that by modulating the surface of nanoparticles with the amine groups, you are able to get the treatment to lung metastasis, and also try to understand if the common knowledge that PEG is on the surface do work, because indeed when you formulate this kind of system, PEG is expected to be on the surface, but this cannot be the case. So we try to look at other functionalities to add to our nanoparticles, and we look to angiogenesis, which plays a critical role in tumor progression and formation of metastasis. In general, antiangiogenic therapies have failed, and they are mainly directed to this receptor, VEGFR2, while it has been reported that VEGFR1 is another receptor which is very much involved in angiogenic switch. And there are some literature data indicating that small peptides can be very useful to block this receptor. And this is the selection we made with this hexapeptide that is reported to inhibit this receptor. Another aspect is related to targeting. As I mentioned before, modification of the surface with ligands overexpressed in some tumors can be very useful to improve the activity of the treatment. And we looked at this receptor, it's folate receptor alpha, which is overexpressed in several epithelial tumors and mainly ovarian cancer. Indeed, we had demonstrated that folate target nanoparticles can be formulated. It is not evident that the folate is placed on the surface of the system, because folate is very lipophilic and it has a tendency to be inside nanoparticle rather than exposed on the surface. So we synthesize these, I call them building blocks of the system. So amphiphilic block copolymer of PEG, we modify the end group covalently through attachment of this anti-FLT1 antiangiogenic peptide, and these other blocks consist in the end group modified with folate. So we had the possibility to build this nano platform with docetaxel inside, and in the first part of the talk I will show you some results obtained on the nanoparticles modified only with anti-FLT1. So these are the properties of the nanoparticles. You see here the size below 100 nanometer and a negative surface. And what you see here is that when you modify the end group of PEG, you immediately have an impact of the on the thickness aqueous layer you have on the top of the nanoparticles. Again, you see that independently of the modification of the system, a sustained release up to 72 hours occurs. These are antiangiogenic properties of this docetaxel-loaded nanoparticle. Docetaxel is a drug endowed of such antiangiogenic activity, which occurs at non-cytotoxic concentration for these endothelial cells. In a conventional tube assay, you see here that when you entrap docetaxel in fully pegylated nanoparticle, a decrease of antiangiogenic potential occurs, while when you have, these are the bars in green, this anti-FLT1 on the surface, you have a higher inhibition versus control of the system, which means that we have higher antiangiogenic properties as compared to docetaxel by himself and as compared to DTX, DBL, unmodified nanoparticles. So we were interested to understand if the system could work also in a more complex model So we employed CAM, which in general is a good model of angiogenesis. But this time we tried also to take advantage of the fact that cancer human grafts can be installed in CAM, and the idea was to develop the embryo, then to graft the cells, and then to treat the system. Tumor starts growing, and then you can stop some embryos, sacrifice some embryos to detect angiogenesis, and then to follow other embryos in order to evaluate the rate of tumor growth. And this was done in collaboration with a company in France, which is called Innovation. These are the results of the in vivo angiogenesis. You see here that also in an in vivo setting, a decrease of the number of vessels per tumor occurs when using these nanoparticles modified with anti-FLT1. And the results in terms of tumor growth are very good. You see here that a significant decrease of tumor weight occurs when the system is decorated with anti-FLT1. No effect was seen on metastasis potential. Indeed, you can also follow the number of cells escaping for the primary deposition site from the upper chamber of the embryo to the lower chamber of the embryo. So the next step was to understand if this approach could work also on a targeted system. And for this reason, you see here, we tested the complete nanoparticles bearing anti-FLT1 and folate on the surface. What you see here is the nanoparticle labeled in green, permeating through a tumor spheroid. In this case, we employed KB cells, which overexpresses folate receptor alpha. In terms of penetration, you see here that it is completely different when the surface of nanoparticles is decorated, especially here. But this translates in a comparable cytotoxicity toward the spheroid treatment, which means that this model is not informative of what was actually what was the actual potential of the system. So we moved to zebrafish in collaboration with University of Padua, biology department. And what you see here is the setup of the experiment. We inject KB cells labeled in red into the yolk, and then after 1 day we inject the treatment, and 8 days post-fertilization we monitor what happens in the tumor. In KB-free embryos, you see here, the treatment was very well tolerated, much more as compared to free DTX. In terms of tumor growth, we see again a higher effect of the system decorated with the peptide, with the antiangiogenic peptide, as compared to the system without the antiangiogenic peptide. So suggesting again that a role of angiogenesis is actually working here. So to sum up our results, it is important to underline that this strategy can be common to other chemotherapeutics, and it is possible to impart these antiangiogenic properties to simple pegylated system. The impact of angiogenesis and the effect on tumor growth should be addressed in different models in order to get reliable information before moving to animal models, of course. And in general, what we see is that a combination of effects can be very useful to extend the applicability of pegylated system. And the last message is that polymeric system offer several advantages, but maybe the best one, the most important, is the fact that through a controlled assembly of polymeric building blocks, you can obtain smart objects. And at the end, I would like to acknowledge all the colleagues at University of Napoli for drug delivery, University of Milan, and colleagues at CNR for the synthesis of polymer, Padua for the from the Department of Biology for biological experiment, and of course funding. And I'm very proud that the Department of Pharmacy at Tonina has been appointed with the Certificate of Excellence in Chemical Science for Research, and will be the recipient of supplementary fundings for the next year. We hope to potentiate also the drug delivery group. And thank you very much for your attention.
Thank you very much, Fabiana. So time for questions now. Any questions from the audience?
Yeah, hi Fabiana, very nice talk. How do you think these kind of structures, well, you know there's all these problems that are being researched right now about the capability of nanoparticles to penetrate inside tumors. How do you think the structures that you are working with can fare, and what do you think might be important parameters about that?
That's a good point, because in general we have a reductionist approach. We look at cells, and sometimes we lose what is on the whole a complex system. So if you think to intravenous administration, you are perfectly right. But from the talks I heard today and yesterday, it seems that maybe intratumoral application is a very reliable option. So maybe we are very much concentrated on this problem of pharmacokinetics, which is common for all drugs. I mean, nanoparticles behave as small drugs, interact with proteins. So this is not a problem for me. But maybe we need to concentrate much more on what is the local environment. And maybe this can be done if also people from at the medical side becomes aware that it is simple to have an intravenous administration, but they can be skilled to treat locally tumors. And we can open through this strategy a number of options for the treatment.
Okay, thank you so much.